He's an extensive commentator on human population genetics and personal genomics. We'll mostly save Q&As at the end unless you have something especially profound. Razib is going to take office hours afterwards in a location that we are both currently unsure about. In the garden, he'll be taking office hours just over there. And otherwise, without further ado,
all right.
Welcome, Razib.
All right, everyone. So I'm going to do thank you. We'll save that for the end. So I want to do a quick survey, actually. This is like extemporaneous, but how many of you have done your genotyping service like 23andMe, Ancestry, et cetera? So raise your hands. Okay, that's a reasonable amount. How many of you have been whole genome sequenced? Okay, that's a reasonable amount. Okay, so you guys know your stuff, and we have some range of people in here. This is going to be, I tried to take manifold markets, forecasting idea and try to make some projections. I've been in this game, frankly, just like bullshitting for 25 years. So I've been wrong and right about various things, and I know that I'm going to be wrong and right about various things that I say here. I'm going to put in some science. If you guys didn't go to the Gene Smith's talk, that was a really great one. Watch the video. Some of the stuff I actually knew about that he was talking about, but I didn't know that they were publicly talking about it yet. So I act like I don't know that, but there is some stuff that's happening. So I just want to mention that. So I'm going to start out with a quote with Craig Venter, who's a really nice guy, but also a megalomaniac. And so, this is the type of quote that he does out, and he, kind of like the big guy, along with Francis Collins, driving the Human Genome Project 25 years ago. So of course he's going to say 21st century will be the century of biology. That's cliche. People have been saying that since I was a little kid back in the dark ages. But genomics as a field obviously is a bit more recent than that, around the year 2000. And I think this isn't totally wrong. I think in biology, genomics is the field that has been getting most of the attention. And so as a service announcement, I'm obviously going to be talking mostly about genetics, because that's my background. And I do think genomics is the sexiest subdiscipline right now. It's synthesizing with technology. It involves AI and all these other things like machine learning, which a lot of you guys are interested in. Neuroscience, when I started out commenting on biology, it seemed like it was going to be a rival, I think, with all the fMRIs and stuff. But I think with all the false positives and everything like that, and the replication crisis, I don't think it's really panned out as a rival yet, and we'll see. I mean, I hope that they get to do things at some point later, but I'm going to mostly talk about genomics in this talk. In terms of who I am, I think a lot of you know, but I always ask ChatGPT and Claude and all these other services what they think of me. I will tell you, do not trust Gemini. Just do not trust Gemini. Just putting it out there. Consistent pattern. I've talked to my friends at Gemini about this, and they're like, hey, it's above our pay grade. So, I am a geneticist. I do identify as a writer now. After 25 years, I've decided to kind of accept this is who I am, partly. I've written for a variety of publications, and I have a podcast, and I also write a lot of essays. I got a couple of essays on Australian Aborigines coming out about their genetics in the next, 1 to 7 days. We'll see. And if you want to learn more about me, I have Razib.com. And if you want to go to the Wikipedia entry and improve it in any way, go ahead. So, obviously when you're doing predictions, as most of you I think know, they're often a vision of the present into the future. And so this is a painting from the Paleo Future, I think from the Paleo Future weblog, but there's a lot of paintings like this. And it's the prediction of 100 years in the future, but it just looks like people at that time with some gadgets. And so this is natural, it's human nature, we're gonna do that. We always do that. And, sometimes though, they are correct. They're not always incorrect with their predictions, even if kind of the stylistic and aesthetic garb is anachronistic, right? So I think like Jules Verne is a good example. When you compare Jules Verne to H.G. Wells, H.G. Wells didn't really try to predict anything. He was a science fiction writer that was just very speculative. And I think he's more remembered partly because he has more literary cachet with his ideas, with his big ideas. Jules Verne, there's arguments whether he even thought himself as a science fiction writer. That term wasn't really in existence. It was like scientific fiction or something. But anyway, he made a bunch of predictions, and a lot of them kind of did pan out, in terms of his undersea narratives, basically depicted a submarine decades before submarines existed. I didn't realize, I think it's From Earth to the Moon or something, one thing he wrote, they launched from Florida because he knew enough that you want to launch closer to the equator because of velocity. And there's forms of radio. Electric cars, as you know, were kind of around that at the end of his life, but I think he wrote about it a little earlier. I didn't know about the scuba gear stuff, but That was interesting to me. So, we aren't just bullshitting. We do land correctly here and there. Some people are strikingly prescient, and so hopefully we will be here. And this is why we have manifold markets and we have prediction markets. We're trying to get a little bit more concrete and hold ourselves accountable, right? And so that's always been a big thing for me. Like, I think about how I'm wrong, and in this community, I think that's a value that we have that's a little different than the normal human psychology, where it's just to try to figure out how we can never admit that we're wrong, because you learn by admitting you're wrong, right? So, basically, I want to admit here that I'm deep into genetics and genomics, but obviously the further I go away from that, I don't know anything, at least compared to like what I know in the fields that I know. And so let's admit where our weaknesses in prediction are going to be. And obviously, if you go 100 years into the future, I mean, a lot of those are just like really, really crazy. Now, on the other hand, if you make a prediction, as I'm sure all of you intuitively know, 10 to 20 years, well, you kind of know the general range and outline because you have the parameters set. There could be a new revolutionary technology that transforms everything, but those don't come around all the time. So when I'm talking about like 10 years in, 20 years in, I think you all know, a lot of you are old enough to know that those predictions do pan out. And I think some of these that I'm going to make will pan out. And, the ones that I made in the early 2000s, some of them did actually, like in terms of like, a lot of you here have done whole genome sequencing. And I remember the first time I was going to rationalist events in like 2008, 2009, I said like, give it about 10 years. And I think that turned out to be right. But, that wasn't a genius prediction. You just extrapolate. When you can extrapolate, it's, easy, right? I wanted to think about this in terms of like what I was reading in 2002. In 2002, Gregory Stark wrote Redesigning Humans, Our Inevitable Genetic Future. And it was kind of a raw, naive book for a lot of people that I talked about it with in the biological community, because there was no way we understood how we're going to do genetic engineering on humans with recombinant DNA technology. That's just like really difficult, and so it wasn't going to work. So we didn't have the technology to do a lot of the things that he said that we were going to do or we were going to try to do. But as you guys know, CRISPR happened. And it's not perfect. It's not really for prime time yet, but we can actually see a future, or I think we can see a future where Redesigning Humans, that book, actually is relevant now. So it took about 10 years, and then it became kind of relevant because there's a new technology. And so sometimes you start with science fiction, and then they become fact, because we can't anticipate innovations. We can't anticipate new technologies that will actually put things into place. But sometimes we can put the framework out there ahead of time and think, well, what if we could do this or that? Because inevitably something happens. A lot of you probably have a biological background. Remember back in the 1980s, before PCR, before polymerase chain reaction, how difficult it was to get DNA. That's why mitochondrial Eve, they got it out of placenta because they needed a lot, and they got it out of mitochondria because they needed a lot of material, right? They didn't have polymerase chain reaction to amplify. That happened in the 90s, and that transformed everything. Then genomics happened, and it transformed everything. And we can talk about paleogenetics, which I'm not going to talk about too much here because this is about the future, not the past. But technological revolutions have transformed basic science and the way we do science. All right, so let's review really quickly, because I like to look at the historical stuff. So there's a bunch of New York Times articles about genetics and genomics, and they're just really funny. So in 1987, New York Times says we have 100,000. Actually, genes, not genomes. That's my mistake. In 2001, the early papers from the Human Genome Project say 30 to 40,000. Now our best estimate is around 20,000, maybe 19 to 20,000. So we keep converging on lower estimates. I think partly it's because we're just looking at us. We're like, oh, we're complicated. We should have 100,000 genes, So that was, I think, what was really going on. And I've talked to older geneticists, and they're like, yeah, there's no way that we could have less than 100,000 genes. But we did, and, it's converged onto this relatively low number, And they were talking about this for decades as if this was a fact. So you can read the articles, and these are the facts that they have. The facts were totally wrong. The world as they understood it was totally wrong, actually. That's how genomics and genetics have transformed in the last 40 years, going from kind of this dark matter of speculation to really, really precise knowledge of many, many things. So we do live in the future, obviously, future from the year 2000. We had 200 human genes. with mapping in 1983. And so now we have all 20,000, 19,500, whatever they say, genes. This was due to shotgun sequencing, as you guys know. You can Google it. It's just one of the things that was important. It was driven by technology, and it was also driven by Craig Venter's ego, which was super important, even though he was very abrasive. He pushed Francis Collins and the government project a lot. The first human genome, its cost was $3 billion, which is a fair amount of money, right? Today, you can get a high-quality human genome of that quality from Nebula for $200, right? And so it went from blue sky to consumer product in a single generation. A lot of you have seen this cost per human genome chart from the NIH, and it shows how for a while it was crashing beyond Moore's law. And a lot of that has to do with the Illumina technology was such a big revolution in the late 2000s and early teens, and now we have an Illumina oligopoly, kind of, or quasi-monopoly. And until we have like a new player pushing them, it's been pretty stable. It's been going really slowly. So I want to tell you guys about what I did in 2014. And I can talk about this now because it's been so far from academia. So what happened was my ex-wife was like telling me, like, are we going to get our kids sequenced before they were born because you were talking to me about that? I'm like, yeah, we can do it. So I started asking around, and I asked this guy, Jay Shanduri at University of Washington. And he was like, no, we're not doing that yet. It's just like too controversial. And I was talking to my PI at the time, my professor, my PhD program, and he was like, well, we could probably do it. And we like figured out, how do we do it? Well, I was working on a mushroom project, and so he was like, well, I can allocate half the sequencing lane from the mushroom project. And he's like, but you need to get the amplified DNA because I can't do human tissue. And so I was like, okay. So we did a genetic screen in the first trimester of my son, and then I asked for chorionic villi sampling to get the tissue to amplify the DNA. And then I called the lab and I was like, I want that DNA. And they're like, we've never gotten this request. We're not going to give you the DNA. And so then I called my friend, the journalist, and I said, can you start tweeting about this? And so she tweeted about it, and then an hour later they called, and they're like, we're going to give you the DNA. Right? So this is all like kind of human social engineering. It has nothing to do with technology, but that's what I had to do. I got the DNA, I put the DNA into the lane, and then I did all the analysis. So that's how my older son became the first human known who was sequenced before he was born. Okay? And so that was like all in the media. And I've asked around, I still haven't found anybody else. And like initially we kept it to ourselves because it wasn't like that big of a deal. But Jay Shanduri asked me, hey, do you know if anyone's done it? And I was like, oh yeah, like stupid. I shouldn't have told him. I was like, oh yeah, I did it 2 months ago. And this is how I did it. And then like the media started calling because like people call Jay and ask him, has anyone done it? And so then he told them my name, So I'm giving you this story just to say that you can be the future in a way if you push it. And I think a lot of you who are entrepreneurs know this, right? So, there are futures we didn't have. This is like a picture of the nuclear future. And I always wonder about this in terms of, is this a future we might have had if it wasn't for the will? And so my hypothesis for this is that with nuclear technology, the capital inputs are so high that social movements like the 1970s environmentalist movement were able to basically cut off funding and social support. And people are just scared of nuclear. People are just terrified of nuclear if you look at the surveys. And so it never really progressed. It kind of stagnated in some, middle population European countries. And we just don't have the nuclear future that you read about in the 1950s and 1960s. It's just not there. If you read the science fiction, if you read like the future predictions, it's all about all this nuclear stuff that never happened. So I worry about that for genetics because people are scared of genetics as well. But the thing about genetics that's great, it's more decentralized. Right? And so you can do it in different places, and I can do ad hoc things myself, other people around can do ad hoc things. So I think it's going to proceed no matter what. There can't be any movement that shuts it down everywhere. I think right now in 2024 we need to think bigger in terms of sequencing. So the UK Biobank just completed the whole genomes of 500,000 individuals, right? So we went from 1 to 500,000 in 24 years. The cost was $250 million. So I just did like the rough math. So that's like 4 trillion if you sequence all 8 billion humans, which is like, I think the Broad Institute's doing like one genome every 4 minutes. I don't even know if we have the capacity. But the world GDP is 100 trillion, so that's 4% of the world's GDP. Apollo mission cost 2.5%. During the years of America's GDP when we were doing it. I don't think we should spend 4% of our GDP on sequencing. But I do think sequencing everybody I would say it's probably more directly relevant and practical than Apollo. So I just want to give some perspective here. And I think it's going to get way cheaper. So it's going to happen, and we're going to have to think about what we're going to do with most of the world being sequenced. And so when is that going to happen? I just did the math in terms of the numbers. Like in like 2008, 2009, you can find an Excel sheet with all of the people that were sequenced, and then it started to get like really, really difficult with private sequencing facilities and other stuff like that in the teens. So I'm estimating like maybe like I think the Broad Institute said they had 100,000 just themselves in 2018. Right, and so they're one of the bigger players. So I'm assuming like right now the All of Us project is saying that they're going to do 1 million. I think there's 1 million out there probably already, and just like at the current rates of sequencing, projecting from say 2010, like by about 2040, it says that most people will be sequenced. I think that this is like overly optimistic. I think we're going to run into a lot of blocks in a lot of different countries, right? And so what are the obstacles? They need labor, to run these Illumina machines. There need to be people around. Back in the 1990s, they were talking about how everyone was going to have to be a systems administrator because of the way technology was exploding. Obviously, that didn't happen. So that's going to change. We don't have the IT infrastructure. My previous startup had to do a lot with that because there's a lot of data being generated, and there's not enough capacity to absorb that. The biobank in the UK has 500,000 genomes, and the stuff that I'm hearing about, I think they're working with DNA Nexus and a couple of other companies, it's really, really difficult to actually use those sequences usefully. And then obviously there's ethical issues related to privacy and reuse of data, and that's a big deal. I don't want to totally dismiss it, but let's just go back from the bottom. I don't really care about the privacy stuff personally. You can actually get my whole genome, from the raw reads to the BAM files to everything you want if you just Google it. Okay, so I mean, unless you're like president of the United States, I don't really see what the big point of privacy is. And like I think like most of the long-term care insurance is already gone because of the information asymmetries, right? So we need to upskill a lot of biologists. I think a lot of you guys probably know that if you're in the field. But I think that's going to happen. And the genome technology is improving. We've been hearing about nanopore, which is like arguably the biggest rival, I think, to Illumina for 15 years. But someday something will happen, and Illumina will not be, the monopoly, and the prices will start dropping. The technology will get better, and the long read revolution will happen. And you're not going to have like a big machine that's like a mainframe doing all the sequencing. Okay, so it's going to get democratized. It's going to get decentralized, and I think it will spread. One thing that people often say, and it's not totally unfair, is that the genome era has been a bust. If you read the interviews, listen to the interviews on Charlie Rose with Francis Collins, it was crazy. Like we were going to cure cancer with a single human genome. I mean, it's just ridiculous. It was ridiculous stuff. But it's not been a total bust. I mean, there are findings like this one. I think it's from a paper in 2019. I think it was a hospital in Minnesota. And so basically, there's a subset of infants that are born that don't flourish. And what they did is, I mean, you can't talk to infants. You can't talk to newborns. They're not going to give you any info. So they sequenced them. And then they found from the ones that they sequenced, they found what was wrong in a substantial number of cases, right? So this is not a trivial issue. So we can do things like sequence people very, very rapidly now, whereas 25 years ago, it took a decade to sequence a combination of a bunch of people that they said was one person at the time, I think. But anyway, you get my point. So I think that, in terms of forecasting, I assume in the next 20 years, like a lot of people will have their genome sequence banked. I don't think the majority of the world's population, but I think it could be like half of America. I think a lot of children's risks will be known from birth. I think opt-out will start to become much more, people are already talking about it in England and other places. I think embryo screening will expand beyond Down syndrome. I mean, there's Down syndrome, the things like Gaucher, there's a couple of other Gaucher's, there's a couple of other bad diseases with deletions and chromosome abnormalities. I think it's going to expand beyond that. And I think the integration between trait and behavior information, right? So imagine a table or information joining the information in Facebook, your behavior on Facebook, your shopping habits, your medical records, what you're doing on TikTok with 23andMe and your whole genome sequence, right? Like for privacy reasons and also like scaring the crap out of people, they're not going to do that. But they could. And 23andMe, I mean, I think Anne has enough money, it doesn't matter. But if it wasn't run by someone that was so highly capitalized, they might sell to some other non-genetic company, and then who knows what they could do, right? So I think this is something we need to think about. We will have this information. The more genome sequences you have and the more information you can combine with it, the more insight and the more value you get. So I always tell people, when you sequence your genome, it's not a depreciating asset. Yeah, you get some information. Maybe you don't get some important information, but as you age, as we get older and older, as we like progress into this century, we know more and more information about it. So you get more results that come out. And like my genotype, my gene sequence has been out there for a while. So I periodically get researchers who email me. They're like, "Oh, I find this about you," and sometimes it's not like super welcome, but whatever. You know, I'm pro information. I'm not gonna lie. I have like a few too many loss of functions, in my opinion. I've seen different estimates, and I've had arguments with friends who work in this area. What percentage of Americans have a Mendelian condition? I'm just going to go with the 10% number that it's round. A lot of my friends say it's lower. I think it depends on how you count deleterious. So this is basically tight coupling between the variant and condition. So this is like classical genetics, where you got a gene, you got a mutation. And I think with these, embryo, like if I want my forecast, within 20 years, embryo screening will be routine. Adults will also be targeted with gene editing. And this genetic information is just going to be like part of the information you have. You know, everyone's going to have it. Most people aren't going to look at it unless they need it. Maybe there'll be some pharmacogenomic stuff. But 10% of Americans are going to have some issues. I have an autosomal dominant condition, by the way. I mean, it's fine. But so I guess I'm one of the 10%. But I'm fine. And so I did an X because the affected daughter, I think like this is where we are. And I think this is where we're going to go. I'm just saying this descriptively. I'm not saying it's positive or negative, obviously. We don't talk about it, I think, in the United States because the professional managerial class people have children late, and they're all doing non-invasive prenatal testing, and they don't want to admit what they're doing because that's something that other people do, okay? So NIPT went from nothing in like 2013 to like almost like amniocentesis and other things disappeared within like 5 years. And it's because it's so good at detecting a lot of these abnormalities. Now, a lot of you know there's base rate issues, et cetera, et cetera. We don't need to get into that. But in Europe where they track this well, it's like a 50% decrease in Down syndrome. Because of older mothers and other things, it's not like obviously 100%. In Italy, for example, it's much the decrease has been much lower. The Nordic countries, there's always stories about how, there's no babies born with Down syndrome in Denmark this year. And it's not because their genes got all better. In Denmark and Iceland and the Nordic countries, they just do not get born. So where does this continue? And I think this is the kind of stuff you guys are interested in. I think people are always, so there's different heritability estimates. There's a range. I just decided to summarize it in the middle from the literature because like it changes periodically. The ones that I care about and are always interesting to me, autism, schizophrenia, and bipolar. They're high. They're highly heritable. Now, that doesn't mean it's what percent is genetic. As you know, a lot of you do. Heritable variation is the proportion of the variation due to genes of the population. But still, a lot of heritable traits, like we can't target their genes. We know which genes they are. We can create a polygenic risk score. And so in terms of autism, schizophrenia, and bipolar, I looked at the unemployment numbers. They go from different countries, and they range all over the place. But a lot of people with autism just don't work. And I've known some of these people. Schizophrenia, I think you guys know why they don't work. Bipolar, I don't know too much about bipolar. I think so. But 50% of adults with bipolar are also unemployed. So these are huge problems. Now imagine you're having children, okay, brass tacks. Some of you will or have. And you're thinking like, you have a history of autism. I have a history of schizophrenia in my family, okay? A lot of people have a history of autism in their family. It's segregating, so the genes are floating around. But there's variation among the embryos, among the siblings. What do? Like, do you take that risk or not? I think we know what most people are going to eventually do. And that's why we're having discussions about it. There's other things in here that are important, like ADHD. I was surprised it was that heritable, actually. I haven't looked at this literature in a while. And then Alzheimer's, there's a lot of variance. Alzheimer's is heterogeneous, right, in terms of what the genetic basis is, but there's a lot of variance. So I think that makes sense that people would target it, but I think that's not a controversial thing, but I don't know. And then let's talk about IQ. We can only explain 15% of the population-wide variation from SNPs right now. There are people that I respect and trust who say we might not be able to get above that for technical reasons. And there's other people who I trust who say that Peter Vischer, he's at Oxford now, the whole genome sequencing will actually bridge the gap like it has with height, and we're going to get much closer much closer values between SNP heritability and like the twin studies heritability, okay? I will tell you, I have my whole pedigree genome sequenced, and I did a rank order. I had a friend of mine just do who does this for a living, do the polygenic risk, and the rank order is exactly what I was expecting. What? Yeah, well, I mean, so anyway, this is what he does. So I was like, I gave him the sequences. I gave him the sequences, and I didn't tell him who it was, and it came back exactly. So my daughter has the highest score in general in my family, and she came back also as the highest. And just the rank order was, so you can get, I mean, I'm not saying everyone's going to be like that, but, I was pretty impressed by that. And so, I mean, people are going to be looking at it. Some of you know that some people are already looking at it. It's probably going to be in the shadows for a while. But, I say like around 20 years, I think it's going to be a real discussion that people are going to be having. Depending on if we can get the SNP heritability higher or not, I think, is a lot of it. So there is a collective action problem here. The genes associated with schizophrenia, and like I have friends who are in behavior genomics, they tell me it's true. It's a real result that there is a correlation with creativity, right? Which not entirely shocking if you know what schizophrenia is like. And then, this is more like anecdotal, but a lot of people, autistic people I'm not saying like I'm in a, okay, I'm not going to say it, but you know what I'm saying. Okay, see. Well, I mean, so, I mean, the issue is like a lot of autistic, like the autistic people that you see like on Rain Man and like the people we know, they're one sort, and that's like, it's like the plane with the bullets. And then there's a lot of autistic people that are like wearing diapers into adulthood, and like I have like, friends who's like, their brother is like this huge autistic guy, and their dad died, and their mom is small, and he's violent. And so they're like, okay, we need to like, what do we do here? Because sometimes he goes through rages and he acts like a toddler. So I'm just saying, like, okay, like that's the one side why people are going to screen. But the other side is like, you know what I'm saying the other side is. So this is a collective action problem. I don't have a solution for it because I'm not going to tell someone that, you have to take this risk for the human race, as an individual. But, we see this in a lot of society, in all of society, where there are these positional games that people are engaging in, and it's ultimately a waste of energy and time. But, are you going to tell that parent not to invest more education or whatever in the children? So people are going to look at these things as investments, And so are you going to tell people that they're not going to invest? I don't know. So I think the prologue of the next 20 years is here. Mendelian screening is going to get better. Polygenic screening is going to come out of the shadows. It's going to get cheaper, more ubiquitous. Ethical issues is going to be like touch and go depending on the culture. Catholic cultures, I think I think the United States, we are individualistic, and I think we will rationalize it. That's what we do. Like, technically we're a very religious country compared to Europe, but we love money, we love success, we love high achievement. And so we're just going to rationalize it. And I have like certain like thoughts that some of you have seen. Like last I was checking the literature, the de novo mutation rate. These are new mutations. Well, I mean, this should vary among embryos as well, right? So imagine that you could like look for embryos with not just de novo, low de novo mutation rates, no mutation rates. Like this is like your genes, but like the best version. That's the dream that I have. And when I was, I don't know how this happened when I was in grad school, but they had me teach the bioethics class as a TA. Again, that's funny, but yeah. They also, the undergrads were like, what the hell? But I was always like, imagine you, but better looking and smarter. Because like they were always opposed to it initially. But I convinced most of them because they like thought about it and they're like, yeah, why not? So, but that's an individual consideration because people are saying, oh, then everyone's going to engage in, positional games. But, our public health is way better. So, it's just like a lot of people don't have hookworm that used to have it. That's great, right? So I think of it as a public health measure, but that's my opinion. So I wrote this thing in 2015, the read-write issue regarding genetics. So we're talking about reading, but we're going to write it, right? We're already writing some of it, but really you read first and then you write in this case. And in 2014, 2015, this was all speculative because CRISPR had just come out. But now it's more mature, but not mature is what I would say. So just to remind you, it's targeted, low error rate, easy to implement. So my friends that work on the bench, I don't work on the bench, it's like they just rave immediately about how easy the CRISPR kits were. It was like a transformation, A lot of labs switched from Talon, Zinc Finger, some of you recognize those things. They've been working on them for decades, and they're just like, fuck it, we're just going to do CRISPR now. It was that good, It was just like getting rid of workflows, the pre-PCR era. It was the same. 2020 Nobel Prize, Dunand Carpentier. There's some arguments about this. They're great. They did the basic science, but I do think the Broad Institute, they really productized it, as what entrepreneurs would say, in terms of scaling it. And this is the literature search that I did. You can see how quickly it changed. If you were in biology in academia in 2012 and 2013, I think you'll feel this. Like people started whispering and talking about it in the halls, and labs started changing protocols really fast. But this was a revolution. This was a revolution that was not anticipated, right? But its market size is still small. I don't know if I believe this, but I looked it up, and it says 35 million, because there's not that much CRISPR therapies out there, right? Many of them have failed. The stocks are down. The companies are running out of money. And the public knows them for the CRISPR babies that He Jiankui did, right? So this last 12 years has not had that, in my opinion, that many great yields. But I think next 12 years, if I had to pick, will. Because revolutionary science, usually overhyped in the short term, but underappreciated in the long term. Those of you old enough to remember the dot-com boom know all the articles in the year 2000 and about, making fun of Amazon. Where's Amazon now? So I think that's CRISPR's trajectory, the hype cycle trajectory, right? There has already been some successes, though. You probably know about sickle cell. There are trials on beta thalassemia, on cystic fibrosis. I think they're doing on dogs because dogs have bigger lungs. ALS, I'm really interested in that because they die of asphyxiation. These are Jerry's kids. That's what you used to call them. Tay-Sachs, which from Ashkenazi Jews, it's painful. Like infants dying of it painful. Now, Ashkenazi Jews screen, but it's found in other communities like Cajuns and Quebecois. There's still kids that have it, right? So this early blindness thing, quick story. I have a friend. He lives in Germany. He wanted to get his kid tested. They're not into genetic testing in Germany because of cultural reasons. And his kids got this. And if they had intervened earlier, because the kid was an infant, couldn't tell, If they had intervened earlier, it would have been much better. He might have had much more of his sight much later. And also there's cancer risks and other things. So I don't know. I don't like to talk to my friend about it because he gets really enraged about it. But anyway, the goal here is not to create marathon runners if you have CF. It's to keep people alive, make them so they can live for a long time. Type 1 diabetes people used to die really early. And now they don't die that early at all. We don't even think about it, right? So I think that's what we're going to see for Mendelian diseases, hopefully, within the next 20, 30 years. In terms of other organisms, I had a friend who was working on goat testicles and making them super fertile, but it was super secret because they were worried that animal rights activists and religious fundamentalists would come after them. So there's stuff that's happening. I don't know all the details, but animals, people get freaked out about. Plants, nobody gets freaked out about. There's still a lot of bureaucracy, though, about GMOs that makes it hard for small labs to actually deploy CRISPR. So I don't know what's going to go on with that. But, I think this isn't going to be revolutionary. But, I think more food is good. So that's a positive, right? And I think that's going to be good. Although, if everyone acts like South Koreans, we're not going to need that much food. There's not going to be people. But, that's the one thing that I would think. Okay, weird stuff. Post-2050. Okay, I'm not going to get into the details of some of the conversations I have, but there's some weird people with weird ideas that are in biology now that are going to be raising money from people here. And, uplift is a science fiction concept. It's been around. Gorillas have big heads. That's a lot of room for a brain. So obviously, our brains can only get so large, et cetera, et cetera. Elephants have big heads too. So I'm just saying, if you humanize elephants, it's not going to be easy, but I think we can see a path towards humanized elephants. And they have, they have the snout. That's good, right? In terms of the apes, I'm thinking North Korean gorilla super soldiers. I don't know. I just don't think people would normally want to do this to apes. I feel the human rights an issue. What? Yeah. Okay, so Gene Smith's talk. He went into this in detail. I knew some of this stuff, but I didn't want to talk about it because I thought it was secret, but it's not. Anyways, this might happen. This iterated embryo, gametogenesis and all this stuff, where you're just selecting at the embryo level. In terms of predictions, though, I've been hearing about this since 2010 as 5 years or 30 years. So I just have very low confidence about this sort of stuff. If it's going to happen, it'll be revolutionary. But I'm a bit more skeptical about this. Let's just put it this way. I think it's probably not going to happen, but I would be excited if it did because I'm evil. Another question that I get is speciation. A lot of you know that a little bit of gene flow between populations equilibrates really fast, just in terms of math. But if Elon is successful in getting us off planet, I think a lot of people will not be traveling between planets as much. And so I think there's a lot of species concepts, and I think people will still be able to get the egg and the sperm together. But really, we will start functionally speciating in terms of not breeding together if we start to look too different. If you talk about people in space or people in Mars, and okay, I just thought there are some freaks out there that would go for it, but that's a different. Sorry, I just started thinking about it. But I think the way that we will do the information gene editing transit is like, I think if they find something interesting in Mars, maybe people on Earth or in the asteroid belt would do it, but it's not that people would move. Does that make sense? So I think like people will transmit the information, the memes, and then we'll like operationalize it in terms of engineering. I want to say something about Colossal, because like they're in Texas partly, and I hear a lot about them, and they have some really great people like Beth Shapiro, like super like top flight scientist. I know some people on their board. I think they're not on their board anymore. They went into their facility and they were like, it's Island of Dr. Moreau shit. So they're lying about what they're doing with the mammoth is all I can conclude. They stonewall journalists. So there are companies that are doing weird things out there. I don't know if they're going to succeed or not. They're partly funded by like CIA like outfit that like funds startups. So I don't know, like could be some military applications too. But yeah, they don't want you to know, from what I can tell, what they're really aiming for. I mean, partly they're just a money machine, but the whole mammoth stuff is PR, okay? Because like journalists who have tried to dig deeper in any other way get like massive pushback. People won't talk. People act really weird. I've also heard similar things. Like Antonio Regalado basically tries to like poke around into China, and they won't talk either. So there's stuff that's going on, and it's not like nuclear weapons where it's like you see it from space, you know? So this is like for the next century. We were talking yesterday about like, is this going to be like the last century of our species, AGI or biological or cybernetic or whatever. So using my evolutionary thinking, like, basically we didn't start having hybridization problems until like 500,000 years. That's a lot of generations, like with Neanderthals and Denisovans. All the modern human races are cross-fertile in a very like normal way genomically, but there's weird things like the FOXP2, the language gene. It went from modern lineages to Neanderthals 200,000 years ago, and it swept through their population. So Neanderthals were changing as well. So there's a lot of evolutionary things that we can figure out by looking at Neanderthals and Denisovans who split from us about 750,000 to 650,000 years ago, and us. Okay, and we can look at the direction of those changes, and we could imagine we could make us more us, like more Homo sapien. I mean, a lot of it, like people will say, it's just going to make us more neotenous. Maybe. A lot of that stuff is a little bit more complicated when you look at how you ascribe characteristics to neoteny and not, and so it's a little bit more complicated. But I think that there is a possibility that our successor species, like descended from us, just like we are descended from like, I don't know, like Homo heidelbergensis, that we will create it, that we can like speed it up, right? And I mean, I don't have that big of a problem with that. Some other people, will have a problem with that. But another idea that some of my friends are into is like cloning John von Neumann. If you read about him, like you understand why some people want to do this. If we do have AGI, this might be the last best hope, But he was buried 67 years ago. He has a daughter that we can get a genome sequence out of. There's recombination there, so it's not the exact, segments are the same. But, paleogeneticists, forensic people would be able to, I think, get a lot of the genome back. And then, some of the stuff that we were talking about with stem cells and embryogenesis and cloning, and if Sergey, who those of you who went to the earlier talk, if he's right about like basically turning humans into like as clonable as mice. Again, there's a lot of conditionalities there. I think that that's feasible, and I think like this is someone that honestly it would be like a boon. I mean, depending on if you like Dr. Strangelove, but no, it would be a boon to bring him back because he solved so many things so easily and effortlessly that I think like he would impact the world in terms of innovation. He, she, whatever, but like whatever. The von Neumann genome, I think, is something special that doesn't come along that often, and I think like that would be a project that I think a lot of people would be interested in investing in. But I'm not sure if it's gonna happen. This is like I'd probably be dead unless like we become immortal or AGI kills us. I don't know. This is not like a near term thing, but we should sequence it. We should like get the genome. I think he's buried in Princeton with a lot of other geniuses. I think there's a project there to be funded. So I guess like another thing I want to bring up, the question of transhumans. Trans rights is a big deal. One thing is, that's gotten caught up in identity politics and late 20th century stuff and gender theory. But I think the question of transhumans is a real question. What do we think is a human? What do you think parents could do with their children? Genetic enhancements, all these other things. There's a reason that people want to do puberty blockers during puberty. Yeah, you could like gene edit as an adult, but what if you want a kid that looks like a cat? You know, should you be allowed to do it? It's your kid. You know, I mean, these are questions. I think most people would be like, no, fucking no. You know, but you know, there's gonna be people who are like, it's my right to create the feline kid that I've always wanted. You know. So I think like this is going to be the real debate in the next couple of decades as we get better and better. This is going to affect the trans debate. I think transgender stuff is going to get better just naturally, just because of demand and technology. I also think, so I realize like diversity, inequality, and exclusion. I think like if we do a lot of gene editing, people's preferences will mean that there'll be like, you know, not Hume, but Burke's 1,000 platoons. People will like prefer their own thing. So if you look at the polygenic selection stuff, you can't do everything, you know? Some people want high IQ, but a lot of people, they're like, why the fuck? Like, I just want a tall, athletic kid. And in fact, that's what like a lot of sperm donation banks, they want tall, athletic kids. So I think you're going to have a lot of different populations that are based on preference, because like there's a finite amount of economic, you know, resources for a given person, and also just like, you know, productivity in the economy to allocate towards something. So I do want to like, you know, I want to bring the Mormon quote where it's like, God became man, and man can become God. And I think a lot of people in this community, you know, they think of AGI as like, oh, we're creating a God. But, you know, I think we have the technology possibly within the next century to create demigods. And I'm pretty skeptical that with our late Pleistocene mind or early Holocene mind that, you know, we're going to be able to keep it all together. I'm not worried about like all the normal things like climate change, like exogenous stuff. I'm just worried about like our software and our hardware. It's like, you know, this is like a 10-year-old iPhone. And I think like it's going to be a little weird without some sorts of modifications within a century, you know, whether it be cybernetic or, you know, biological. I want to also say, if you talk to biologists who are doing biology, a lot of them poo-poo a lot of stuff. So I can tell you in the 1990s, geneticists at Stanford a lot of them didn't believe that the human genome could be mapped. I know this because I know them. In terms of I know older professors who told me when they were postdocs, that was the argument. A lot of people were like, that's not going to happen. Because they know the technology they have. And there's a reason that biology is invaded by physicists, because physicists are egomaniacs and they have big ideas. Biologists like have their heads down and they focus on the problems in front of them a lot of the time. And I think that is a problem because biologists do not think like economists. They do not think like physicists, you know? Now, obviously, people who go into industry and startups are a little different, but in academic biology, I do tend to see kind of a little bit of short-sightedness, which is great because, hey, like, you know, you want to solve this particular form of cancer or whatever. You don't want to think like, what's it going to be like in 200 years, you know? But you need some people like that. And I think that we could get a little bit more of that. But I have felt like that being in the biological community for decades, that you know, there's a little bit of narrow-mindedness out there. So I was gonna give a subhead like the triumph of will, but I decided not to do that. It's not optimal, but I think a lot of this stuff is as I was like reviewing, like so for example, I wanted to tell the story about my son because that was just about my will. Like I knew it could be done. If I hadn't done it, who would have done it? I think somebody would have done it, but it might have been a while. And when people ask me in the media, like, why'd you do it? And I'm just saying, well, because I could. You know? And they were like, well, isn't that a problem? And I'm just like, well, it's just because nobody else has done it. Like, you know, second trimester, like, you know, we could have had an abortion, like scrambled, you know, cut his head off, scrambled his brains. And they're like, okay, whatever, you know. Or like circumcision, you know, like they just do it to kids, you know? And I'm not taking a position on that. But I'm just saying, like, all we did was get information about this kid and the media. And like there was like kind of an IRB thing with my professor that was fake and trumped up. Again, like we followed the rules. There was no human tissue. Right? It was just amplified DNA. But people at Davis were mad that I did that because you shouldn't do that. But I was just like, look, we can do it. And it's not like harmful. And so I think we just need to think about what we want to do and do it. Just execute, right? So I think with that, I'll take questions. How much time do I got? Oh, okay, good. Yeah.
Thank you very much, Razib.
Let's start from the front.
Cool. Thanks for a great talk. You mentioned earlier that you mentioned earlier that a bunch of the first generation of CRISPR therapies failed. Can you say more about like what went wrong, like what was off on the
I think, okay,
hypothesis,
so
why
I'm
they
not like a
didn't work?
molecular person. The easiest, like the kind of like the get out of jail card is like delivery is always a fucking problem. Like in terms of like the transfecting and all these things. It's like the genetic engineering from like back when I like focused on this. Okay, like that was like a solvable thing, but like how do you get it into all the cells? You know, was always an issue. So I think like that's the big thing. Delivery. Obviously, like if you're talking about like cystic fibrosis, you know, you don't need that much of the lung tissue back. You just need enough, you know? And so that's why like it's not insoluble. But I think the sickle cell, and there's some beta thalassemia people. Oh, I saw a trial. I didn't know about this. There's a trial, I think, of a kid with Tay-Sachs, and 3 years on, he's good. So I don't. That wasn't like that's not been widely publicized, has it? Yeah, I was shocked by that. Do you know about that, Sri? Okay, so he's in industry. If he doesn't know, it's not widely publicized. I don't know why. People probably don't want to get people's hopes up, you know? But yeah, I think a lot of it's delivery. Also, like, you know, as you guys know, some of the CRISPR treatments were causing cancer because they're like off-target effects, like, you know, they're mutating things. That's bad. That's going to get better. Unfortunately, though, experimenting on humans is a problem, you know? But okay, next. Who's next?
More questions.
Sure, we have their whole genomes. That would be like pretty tedious. I think what we need to do is like maybe target the functional regions and Neanderthalize somebody. But again, that's an ethical question. Like, what if they're really good at math? Anyway.
So, a little inside baseball, I guess, but you said FOXP2 is the language gene. I heard that was controversial. Do you think it is?
It's necessary but not sufficient. There's because there's a bunch of versions. All right, so it's a vocalization gene. It's in birds too, and when they put it in mice, like the humanized version, the mice start singing weird. You know, they start chirping. So there's a bunch of versions, and there's a bunch of segregating versions in modern humans as well. Okay, there are families that have like mutations that are not totally pathological that seem a little weird. So.
Is there speech difference?
Yeah. Well, they're not. Their verbal IQ is lower. It's just not a big deal. People don't focus on it too much. I don't know why. They wanted like it to be everything. Can we get people in the back? Yeah.
What do you think about the relationship between this and advances in AI? Because my impression
Yeah.
of the vibe right now in like lab automation is that you know, a lot of modern, especially drug discovery, but a lot of biotech
Yeah.
in general is like, let's just try, you know, 10,000 variations on
Yeah.
a thing, and the robotics is there. And so if we start using AI to like analyze that data, then we eventually have a thing that basically can just iterate on itself and like do the wet lab science
Yeah.
and then predict the next experiment.
Yeah, there's labs like that. There's, like, a friend of mine runs this thing, Emerald Labs. It's like a cloud lab. But anyway, so I think AI is essential. Machine learning, all these things are essential in biology because we have too much data. You know, I can tell you the genes I love, but like I don't know like more than a couple of hundred genes. You know, maybe like that should be a spelling bee thing. But anyway, like, there's gonna be people who's gonna memorize all 19,000. But like it's like, you know, back in the day, people used to use Excel and like work on like little sequences. Like Sri remembers this, like little sequences that you manually align. And now it's like, you know, get like terabytes of data. You have a whole genome of 3 billion base pairs. If you want to cut it down, you have like 5 million SNPs in a VCF. This is just way too much for humans to handle. So we need to like be really heavy on like this analysis. And so AI is essential. It's going to supercharge our knowledge. I do think that there is still low-hanging fruit in there as we get the bigger data and like more and more individuals get sequenced. I think there's a lot of private variation, and we'll see. Like rare variants, like, you know, these have been discussed. But I think it's synergistic.
Razib, I wanted to challenge you on how big a deal polygenic selection is going to be in the short, medium term. It seems like your view is there are many different traits that people are going to want to select on. Something that people think about here is, you know, optimizing for two things is often a bad idea. You don't really get the benefit of optimizing for either. It seems like if you're optimizing for, you know, if you're doing some linear combination of 10 different polygenic scores for quite different traits that have low underlying correlations, you're actually not selecting very strongly for anything. You need multiple rounds of this selection or many different embryos to really get some power there.
Oh yeah, so I mean, I honestly, I think the thing with the polygenic selection is like I really only care about IQ because the high IQ people are the ones that are going to do innovation and drives things, you know. So that's what I'm focused on. That's only going to be a small minority of the population, but you know, I think if enough people do it, it's going to make a difference, right?
Yes, I mean, I guess I'm asking quantitatively how big a difference that's going to be. It seems like if people are selecting on height as well, it's going to be a considerably smaller
I see. I
difference
think
than like otherwise.
I think some people will. I think people who are like very like on the taller end will. And I think this is what I'm saying about preference heterogeneity. A lot of people, so for example, in this community of like, embryo selection, all this stuff, they are shocked at how shallow normies are. Like we're talking like a rich CPA or something. And they're like, how do I get a hot son? Like, well, are you sure you don't want a smart son? It's like, oh, whatever, So I think like that's one of the things that's interesting. So for a lot of people, they're just going to be satisfied with height, and all that stuff. But I think the minority of people will focus really hard. I mean, I don't know if the iterated embryo thing, it's been talked about for like 20 years now. I don't know if it's going to happen anytime soon. So I think like just normal selection, depending on how many points. Different companies already offer or claim different values, but it's going to get better, right? And I think, 2/3 of a standard deviation, I mean, come on. I mean, have you talked to people that are like above and below? You know what I'm saying?
2/3 of standard deviation is a very big deal, but not if you're not getting that.
Yeah. Well, we'll see.
More questions?
Yeah, anybody in the back? No? We good? No? All right.
Can we pass the mic?
So you talked about the iterated embryo selection, and I've read a little bit about that, but what do you think are the biggest barriers to that?
I don't know, because like I'm just saying that because the schematics are always like nice, and then when they implement it, I think like what is it like the super baby guys who said the genomic imprinting issue with the sex, like that's fucked up. Yeah, that's gonna fuck things up. But the reality is like developmental genetics and biology is very complicated. And for me, like molecular biology is like it starts at A, it ends in Z, and there's a lot of like things that break in the middle. And just my expectation is there's going to be a lot of like I think they call it teratologies, monsters. Like, are you willing to tolerate that? Some people will be. Some governments will be. I think a lot of Americans don't want to see that stuff. So there's going to be like enormous number of unanticipated molecular mechanisms that they're fucking around with inadvertently, even though like it looks really, first of all, it looks really nice in mice, but like what the fuck are mice telling you? You don't even know if those mice are as normal as you think, because like how many traits are you like measuring in a mouse? Anyway, so I think about things like that. People can be like, Dolly turned out to be a little off, so I think, do you want to take that experiment with kids? Look, I'm obviously like open to a lot of things, but like I'm getting creeped out right now, so I think that should tell you like how it's going to be with the public, right? So I think behind, yeah, you've been.
Could you pass the mic backwards?
Yeah.
So the focus of the talk was, the future of humanity. And
Can you speak up a little?
Yeah, the focus of the talk was gene editing and the future of humanity.
A little bit.
Screening. In other areas of biology, what do you have like hot takes?
Yeah, in terms of like, I don't know much about what's going on in neuroscience right now, but actually like you guys know SSRIs, all these other things that people are doing. At some point, people are going. Okay, this is weird, but I'm gonna say it because this is like an audience. Okay, I've talked to a lot of guys that were like, I've talked to them and like, maybe I shouldn't. I'm gonna say it. If I could take a drug that made me gay for a day, I would try it just to see. So here's the thing: is like, what if they liked it, and they never went back? Just a thought. Now, of course, a lot of us in here are gonna be like, we would go back. Would you? So there you go. Because if you can modulate your own preferences, if neuroscience gets good enough where you can actually start rewriting your brain, that's going to be really weird, yeah. So I think, what's up, Omar?
I don't know if you covered this. I actually stepped out, but you said that there could be trade-offs in what you genetically engineer, and I wanted to, I guess, maybe push.
Yeah, I kind of. So I asked this at the other, there's something called antagonistic pleiotropy, where it's like when you select like genes and like extreme distribution. So like the chicken with the big breast that can't breed anymore, is a classic case. Sterility in a lot of organisms when you select on traits. I don't see anything. So height, for example, does have antagonistic pleiotropy. If you're above 6'5" and you're thin and you're a male, there's a high risk of lung tear. Your body doesn't scale well. So above about 6'2" is when physiological fitness starts to like take a dip, and above 6'5" it's noticeable. So being tall is supposedly good, and it kind of is like socially, but obviously there's a limit. When you have a normal distribution in a trait that's like quite heritable, that usually means the selection is not unidirectional, that there's disruptive balancing selection going on or something like that. So when you have a normal distribution in intelligence, you have a normal distribution in height, whatever, that means it's more complicated than just good to be tall, or good to be smart. So for example, you can imagine, polygenic scores for height vary between populations, and they vary in ways that are consistent with what you see in the populations. And like, why would that be? Well, like smaller people have fewer calories, So imagine a situation where the floor of calories due to like drought or something is lower. There's all sorts of reasons like that. But anyway, with intelligence, John von Neumann was a weird guy. Like all of the biographies indicate he's really weird.
Sorry, so that statement that you made was more in reference towards extremes, but there's nothing stopping you from doing +1 sigma across the board for things.
Oh, I have no problem with that.
Yeah, that's
like, I mean, if the whole human population was 115 midwit, I would be happy.
No, I'm not saying like I'm not asking about the desirability, but the technical feasibility.
I don't think it's any problem. I'm not worried about that because they're all common variants. They're all common variants, and we've done the experiment.
There's so many like.
Yeah, but it's like what happens when you move many standard deviations. Are we getting kicked out? When you move many standard deviations is that you start to get allele combinations that you never see in nature, and that's why you get the different expected value. And that's where like epistasis and other things perhaps start kicking in, and the additive linear model breaks down. That's what I would say.
Okay.
I think that is all we have time for. Please join me in thanking Razib.